So it's a really pleasure to welcome these two leading figures and colleagues and friends in the species research and invasive alien species research and management at European and global levels. So Quentin Groom is a researcher, and correct me if I say something wrong, a researcher at Meise Botanic Garden, and an internationally recognized expert in biodiversity, taxonomy, and open data, with a strong focus on building infrastructures and tools that connect scientific data with policy and management needs. I will also present Professor Ellen Roy. She is a senior researcher at UK Centre for Ecology and Hydrology and a highly influential voice at Science Policy Society Interface, particularly in early warning systems, citizen science, and evidence-based decision support. So we are delighted to have you here with us during these two sessions, and we would like to thank you very much your presence here at CIBIO. So we will start with Quentin Groom.
Yes, hopefully everyone can hear me now, you think? Okay, yes, and thank you, Joanna, for inviting us. It's a real pleasure to be here. So I gave a few weeks ago the title of this and I realized when I was starting to prepare it how ambitious a title it was because it's such a massive area. But I'll give it a go and hopefully it sort of stimulates some ideas in you and some thoughts, and hopefully we can have a little discussion once I've finished as well before we get into Helen's talk, which I think will have a lot of common themes, I suspect. So we have this giant paradox. We're creating biodiversity data at a faster rate digitally for more of the world than ever before, about more species and more places and everything. But we still struggle with the timeliness of evidence for policy, how repeatable that evidence is. So are indicators the same as the indicators we did last year, for instance, and how defensible that evidence is. So can we trace it back to the source data? Are the evidence useful, and things like this. And so why is this so difficult? I'm probably not going to cover the whole subject at all, but what I wanted to do is reflect on some of the projects that we've got currently running at Meise and give you my reflections on those. And there are four main projects. One is One Stop, which I co-lead with Helen on invasive species, and I know some of you know that one, looking at how we can use data to join the various different strands of invasion biology together, like detecting, modeling, prioritizing, things like this, and join it together in a much more rapid way with workflows. I have the Biodiversity Meets Data project, which I'm a partner in, which is led by Niels Reyes from Naturalis, and that's about giving tools to conservationists who maintain conservation areas and giving them tools for modeling and things like this that are more useful to them to help them do their job. The TETTRIs project, which is led by the Consortium of European Taxonomic Facilities, which is all about building capacity on taxonomy and identification within Europe. And the B-Cubed project, which I coordinate, which is very much a data science project, looking at how we can use data cubes to make much more rapid analysis of biodiversity data from GBIF. So what I'm doing is just reflecting on these projects, the common themes within these projects, and what these projects can tell us about how we can potentially improve the rate and the quality of the evidence that we produce for policymakers. I said we're getting lots and lots of data. On GBIF at the moment, there is 3.6 billion observations. That's only the presence observations. There are another 100 and something million absence observations on GBIF, and still we don't have enough. We definitely need many more observations. Those come from 120,000 different data sets and 3,000 more publishers. And one thing that always frustrates me is when I hear about people talking about GBIF data, because there is no such thing as GBIF data. Each one of those data sets is different. It has a different methodology behind it. There are eDNA data sets on there. There are citizen science data sets on there. There are data extracted from the literature. There are data from camera traps, all sorts of things. And so you can't compare, you often get the person saying, well, my data set's way better than the GBIF data set. And yeah, well, yeah, which data set on GBIF are you talking about? And yeah, sure, you probably collected it for one specific purpose. So data are not evidence. Evidence has to be built from the data, and that's a process we have to go through. So the evidence also depends on the question. We have lots of different kinds of question requiring different kinds of data. So in one stop, we're very interested in early warning for invasive species. We're also very interested in doing risk assessments for invasive species. And even those two things need kind of different data. We're interested in trends to know what's increasing or decreasing for either invasions or for conservation. The conservationists need other kind of data to know where things are, how to conserve them, what species they may interact with. And taxonomists need different kinds of data to describe the biodiversity of the world as well. And ecosystem services people need quite spatially explicit data to try and quantify those ecosystem services as well. So yeah. So quality of a data set is not absolute. Data sets are collected for different reasons, under different constraints, with different assumptions behind them. So we're often disappointed by data because we expect them to answer they were never expected to answer. So I think standardized monitoring is great. We need more standardized monitoring. We see a trend in Europe towards more more standardized monitoring. When I was last year, Henrique Pereiras was giving a talk about EuropaBon, and there's definitely a push within the EuropaBon and within Europe itself to have more standardized monitoring. We definitely do need that. But standardized monitoring is never going to be over a long enough period. It just costs too much, and you can't do things retrospectively either. It's never going to be broader scale enough because there's just not enough resources. Even if you could spread standardized monitoring across the whole world, there's going to be lots of gaps between all that. And it's never going to be comprehensive enough. You're never going to be able to cover all of taxonomy. So yes, more standardized monitoring, fantastic, but we have to also make use of all of this heterogeneous data as well. So I work at a herbarium. We have very good taxonomic coverage of the whole world, very poor temporal and spatial coverage in general, but we have one of everything kind of thing, or a few of everything, at least for vascular plants and fungi. We have lots of opportunistic observations. I've heard people complaining that GBIF is a data set of birds because we have so much bird data. I can't see why anyone would complain of that. That's amazing. If you're a bird person, even more amazing than if you're a plant person. But if there is something that's easy to observe and we have lots of those data, yeah, let's use it. And that's fantastic. Birds are important. So lots of those data are citizen science, but also we have data on things like remote sensing, which is diabolically for taxonomic resolution, but it's fantastic for spatial resolution. And so if us biologists could get together with our taxonomically resolved data with the remote sensing people, we could do some really amazing things, I think. And that, to some extent, is why B-Cubed exists, to try and put those two sets of data together. So B-Cubed is trying to make something of all of this heterogeneous data. We assess the fitness for use of those data as we feed things into our workflows. We try and make the uncertainties and biases in the data explicit. That's not easy. And we try and reuse data. We're reusing the observations in the first place, but also all the code is made open and potentially reusable by anybody as well. So a data cube isn't particularly complicated. Maybe makes it sound more than it is. It's basically a flat file with taxonomy in it, what species it is, for example, what date the observation was made on or span of dates, and what a grid cell it's in. We tend to do everything in cells to be able to aggregate in cells. You often see people doing species distribution modeling and they say, well, we take the raw data. What almost the first step is to grid it, because all of your environmental layers are in grids already. So often we use things like the European Environmental Agency grids, but there are other kinds of grids. There's grids for the equal area grids for the whole globe and things like this. But every environmental area is in some sort of grid or another. This window here is what GBIF downloads looked like before. Maybe you know those green buttons. But what the B-Cubed project, and GBIF is a partner in the B-Cubed project, did they put on this download as a cube there. And if you try downloading data, you can do this yourself afterwards. Once you click on that green cube, it gives you all the details by what you can aggregate by, what grid you want to use, whether you want to aggregate by year, by month, or by day. And you can create your own cubes. You've always been able to do that with GBIF, but what you would have to do is just offload all of the raw data, stick it on your local computer, maybe put it into a database you own, then do all the aggregation yourself. This shortcuts this. It's all done on the cloud on the GBIF architecture. This takes 15 minutes to half an hour to get your result. If you try and do this yourself, it'll take you a couple of days, because they can expand the number of cores on their cloud infrastructure to be able to do it super quickly, and you just can't do that yourself. And there's a lot of manual processing yourself. This is the way you download also manually, but this can all be done to an API. So this can all be scripted. And so you can run your workflow every single day without you having to touch a button as well, if you just set it all up. So those are data cubes. If you look behind the scenes, and you may or may not, it depends what sort of IT person you are, it's run using a structured query language query to get those out. And I wanted to just show you this. If you've never seen one of these before, it's actually much more simple than it looks. There are 4 sections to this. There's the select, which tells you which columns to select out of the database. There's a from that tells you which database it is, and that you only have the choice of occurrence in this. There's the where, which is the filter. So it's the filter which taxonomy you want to take out, which country the data, if you want to filter by country, which here there's a line, a not occurrence on the basis of record of fossil specimens and living specimens. So we don't want those in our cube, for example. Living specimens are the sorts of things you find in zoos and botanic gardens. And then the group by is, what you group by. And you can see this is being grouped by this here. I didn't put that in red, but it's also the same thing here. This is the quarter degree grid cube for the grid system used in this particular one. And what this does particularly is takes into account the coordinate uncertainty in meters, because almost every record in GBIF has a radius of uncertainty around it. This is what most people doing species distribution modeling completely ignore. They usually just take the centroid, assign it to the grid there, which might not make much difference. But these uncertainty in meters can, if you put something in iNaturalist, it might be a few meters. But if the data comes from an atlas, then the coordinate uncertainty in meters could be several kilometers. And so we try and take this into account when we're making these grids, making the cubes. So anyway, this can all be scripted, or you can do this off GBIF itself. So In this talk, I've got 4 themes. Actually, that was the first theme. That's the conclusion of that first theme. You might not like the data you have, but if you want evidence, tough. This is what you have to use, and you have to figure out ways to do this. So the second theme is most biodiversity data unstructured, which is related to this, the first theme as well. So these unstructured data are more comprehensive, as I said before. They cover a much broader spatial area. They're for much longer time spans, in actual fact, going back sort of 200 years in reality. And we cover many more taxa doing that. It was also a theme that came up last week when we were having the one-stop annual meeting, the difference between bias and uncertainty. I'm not exactly sure of the terminology here, but let's say in this context, uncertainty is a statistical uncertainty, so your standard deviation effectively, and you can quantify that. So we make models, and almost in every case, we have some sort of deviation on that model, and we can plot that statistical uncertainty. But that doesn't deal with the bias. And if you look at species distribution models, we actually don't ever, I don't think I've ever seen anyone trying to show the biases behind the data. And we have a lot of biases. And part of the problem is because we don't have any data that's not biased, even to some extent systematic surveys, that it's very difficult to quantify those biases. We can only try and potentially include those in our models, some of the biases like survey effort, to try and account for it. So this is a paper that I contributed to by Amy Davis from the TRIAS project that was a few years ago. And this is a map of uncertainty for the models. Now we're using this workflow, the Wisdom workflow, currently in the One Stop project as well, and also the Guardius project. Joao, he's not here, but he may be online, but he's also using this. And this is a map of the uncertainty of our models under 4 climate change scenarios for Vaccinium corymbosum, which is a blueberry bush from North America. So it looks as if it's going to take over the whole of, well, that's not the distribution, but the distribution maps I don't show kind of show it taking over the whole of Belgium, which I'm pretty sure it never is going to. But this is a climate suitability map, and those are the uncertainties on there. But it doesn't show anything about those biases. So I know for a fact that there's lots of records from iNaturalist in the USA of Vaccinium corymbosum. But as you get further north, I know the species still existing to Canada, but as you go further north in Canada, there are fewer and fewer people. And so you get fewer and fewer records of Vaccinium corymbosum, inevitably. And so that is biasing these data on the distribution in Europe. And yet it's really hard to communicate that to policymakers once you produce your evidence. They see that. They see it at incredibly fine resolution, which I think is a little bit misleading. And we have completely hidden all that bias in the underlying data on how it matches the climate. And I don't have a good way of getting around this. It is a statistical problem, but it is one of the problems we need to try and address to have good evidence as well for policymaking. So this is just another example. You presumably recognize that part of the world. There's Flanders, that sort of band of data there. This blob here is Luxembourg. Luxembourg borders on Belgium as well, but unfortunately the other half of Belgium is fairly low in data because they don't publish their main data set to GBIF. There's the UK, where all of that patchiness is nothing. Well, these are vascular plants. I should have perhaps said that, but that's just pictures of vascular plants. That patchiness is nothing to do with the patchiness of vascular plants in the UK. It's to do with the openness of different counties. So that's Cornwall there. Next to it, there's part of Devon. Yeah, there's Devon. No, that's part of Devon. I have no idea. Actually, is that 2 halves of Cornwall there? Yeah, so that's 2 record centres in Cornwall, which have different levels of openness. That's Devon. You can see Norfolk is quite open. There's some atlas data there that's been released, and that's why you have all that checkup pattern there. You can see the Netherlands is quite open. You see lots of atlas data there for northern France. That must be around Paris. You can see the border along here of Germany, France, and the Netherlands. You have a weird hexagonal thing going on there. I have no idea what that is. It may be something to do with the grid system. But yeah, again, all you see is bias here. You see no information about vascular plants whatsoever. And it's very unrandom. So it's very hard to deal with statistically because of that lack of randomness. So making uncertainty explicit. Yes, we already do that for statistical uncertainty, but not for bias. We can address fitness for use of the records, whether they really help, whether those records are suitable for what we need them for. We need to output uncertainty, as I think most of us already do as scientists. That's normal, but we're not making the biases behind the data explicit. And rapid and repeatable workflows do help us do that, because if you do a one-off analysis, you can't very easily incorporate new sets of data in there and keep running it and running it and running it, which is what we really need to do. So the end of that second theme, unstructured data are not the problem. The problem is pretending we have like structured monitoring data. We need to really treat these differently. So my third theme. So this is where I put my I'm not a social scientist, but I put a social scientist cap on, because we're thinking about the people who collect the data and use the data as well. And so this was a diagram from another TRIAS paper from a few years back, which was about citizen science, but I think it has a broader message in it, that we have all of these different groups which have an interest in biodiversity data, whether that's collecting biodiversity data, analyzing biodiversity data, or making decisions on biodiversity. And this was the sort of pointing out the fact that these people all need stuff from the data they collect, or fund to get collected, but they also produce data. And so they are all connected in a way together, but fairly loosely. So we have within those communities, there's people who are data collectors, and some people, of course, can be all three of these or just two of these. We have data collectors, and they may be fairly local in their view. They might be trying to contribute because of wanting to contribute to something bigger. They might just be doing it as a hobby, but they might also have some element of local stewardship towards their local environment and why they collect data. You have people, I also have this sort of hat on as an analysis of data. We're motivated by new and fancy methods to be able to do this, and discovering things that haven't been discovered before. And down to the nature of our job, we have to publish. And so we are motivated by being able to publish and publish and publish. And you have the policymakers with all their motivations. They need sort of things done quickly. They have accountability to other people that are not scientists, and they had to make hard decisions on things that are not just decisions about biology and conservation or invasive species, but lots of other things to do with sort of employment and health and things like this. So they have many different motivators on them as well. So each group has a rationale in its own context, with all sorts of different timescales and funding models on that. As scientists, we're always complaining about our short-term funding, but in reality, when you think about it, everybody has short-term funding. The policymakers on their election cycles is usually sort of 4 to 6 years or something. They are all, those civil servants that are paid by the elected officials, also stuck by short-term funding, and that could be just pulled out from underneath them very easily as well. An ecological change happens at fast and slow rates, but nothing to do with biologists' cycles or election cycles or what have you. So we have to deal with that. It adds to that heterogeneity of data It's just something we have to live with, and we may complain a lot, but yeah, we probably should think more about how we're going to deal with that rather than how we're going to stop short-term funding, because I don't think we're going to be able to stop short-term funding. So I get exercised a bit about, there's a lot of talk about sort of indigenous people's rights and things like this, but I think, especially on a European context, there's a much broader issue of local communities that often get forgotten about. And there's lots of polarization. I imagine you have it in Portugal as well, between rural people and the city people, and their different wants out of biodiversity. And those local people have a lot of knowledge on biodiversity locally, and they also have questions on biodiversity, and they may want to conserve things, but they also have to make a living from biodiversity or from forestry or from farming and things like this. And so they have different drivers on their needs from information on biodiversity. So they have local responsibilities, but they often have very limited analytical capacity as well to be able to really analyze those data. So you see so much, I don't know if you have the word hegemony in Portuguese, but basically a large group having a lot of power over a smaller group, or just really a powerful group having a say over a smaller group. And that often happens between small communities. So the reason I started thinking about this is particularly my work on the island of Montserrat, which is a small island with difficulty of having a large volcano that's active there, hurricanes every now and then, and there's only 5,000 people on the island, so they have very little capacity to analyze the data as well. So they kind of need people externally to help them with their data. They have some very clever people there, but they don't have a university, and they don't have a research center or anything like this. So they need support, but I would like to see my help with them as being driven by what their needs are, and finding a way that we can both benefit out of me working with them. So we are thinking about how we can build capacity on the island to do things without being exploitative of them. So they are not sort of just working for us to collect data, but they're working for themselves to collect data for us, and we're helping them be able to understand it. So having shared ownership of questions is important. They're very important to us, that we have questions. Maybe it's about forestry or something like this. So they want to know what's in their forests, what sort of trees there are, so they can potentially exploit them commercially and do that in a sustainable manner. We may also be interested in their forests because they contain wonderful species and conservation-worthy species and things like this. And they're also interested in them, but they've also somehow got to survive on this small island as well. So we also should be looking at how we can make tools so that analysis doesn't always have to go off island, so that there are tools online, for instance, that they can use and answer questions locally and make things a bit easier for them. And we, as scientists, also have to respect the constraints that they give us about what we are or are not allowed to do on the island. So I think all of those things are exactly the same as the things why they had the care principles for indigenous people. But I think that's why I'd rather think of it in a broader sense of the word, because it's not just indigenous peoples that have been exploited in the past. Small communities also have very similar problems. So we did an analysis, for instance, of the specimens that have been collected by scientists. They're in herbarias and museums around the world. And we looked what had been collected on Montserrat and in the Cayman Islands, and where and who is publishing papers on that. So this sort of comes out of the Nagoya Protocol. So in the Nagoya Protocol, you're trying to share the benefits of biodiversity data. You have monetary benefits and you have non-monetary benefits. Now, for a herbarium like ours, where we digitized our whole collection, there's only costs there's very few monetary benefits we can easily put our fingers on, because we don't make profit out of the digitized data, we just make it open. But I assume we have non-monetary benefits. So having a big collection in Belgium means that we have people coming to Belgium to visit the collection. We can make paper, we can write papers based on what's in our collection, things like this. And so those are non-monetary benefits. And as you can see here, in terms of who's publishing on data, using data from Montserrat and the Caribbean, obviously the United States is the major beneficiary of those non-monetary benefits. Also, Northern Europe as well is not doing too badly. Apparently not Portugal, but there's definitely a thing about population here. Obviously, you have a bigger population, you're going to publish more papers. But it's interesting to see that Brazil and China are using the data as well. One reason it will be Brazil particularly is because obviously they share some of the same flora and fauna as the Caribbean as well. So that's one of the reasons I expect Brazil is more up there. But China, I have no idea why. Often the data just being used, it might be just one specimen being used in a much larger study, say on a family or at least. Yeah, you good? It could be, yeah. Yeah. So why traditional models fail here? So this is coming back to the theme of structured surveys. So as scientists, what we'd love to be able to do is set up structured surveys, say for the whole of Europe. So vegetation plots for the whole of Europe in a sort of segmented way, so we could monitor a long-term different kinds of habitats and things like this. But if you try to impose that right across Europe, and particularly trying to include NGOs, I think you have a problem, because you're not considering the communities. And if you try and do it in a very top-down way, and I've seen this happen with the organizations I've been organized, people just go away and ignore you and go and do their own thing if you're too top-down. So this is sort of that same diagram again, but this is from the One Stop project and includes the living labs at the center here. So many of you, I assume, are involved in the Portuguese living lab one? No, some of you. I know Joanna is, that's why I'm looking at her. But so in One Stop, we have 5 living labs. One is in Portugal, one is in the UK. Actually, I don't know, you're not so involved in that now. There's one in Belgium, but there's also one in Finland and one in Romania. So we try and cover lots of different types of area within Europe. And this tries to implement this local communities aspect to it. So instead of flying a whole bunch of people to Brussels who all speak English so that we can have a discussion about a particular subject in Brussels, these living labs work in their own countries, in their own languages. We can then, of course, have people that don't speak English contributing to One Stop. And we use this as sort of co-creational space so that we show them and let them have hands-on use of our innovations from One Stop, and they can feedback whether they think they'll use it, is it useful to them, how does it relate to their local context, and things like this. And so I'm very happy. We've only been a year in, but the feedback we've had from the living labs is very positive, that people have fun when they come along. They meet people from their own area that they wouldn't have otherwise met, and they can discuss local issues. So we don't try and impose the agenda too strongly on the living labs, so they can talk about local issues as well. And yeah, I'm very pleased how that's working. If anyone has been to the living labs in Portugal, I'd love to hear about your experience here. I tend to just hear about the Belgian experience. And funnily enough, I've never been to the Belgian ones because I don't speak Flemish, and it's all done in Flemish. So yes, evidence improves when communities remain distinct but deliberately connected. So we are trying to connect all those communities together with data and put them together in the same room occasionally. So the challenge is not a lack of expertise, but aligning expertise across communities that were never designed to work together. So final theme, capacity and governance and the future. So this relates to the TETTRIs project. So one part of our work at Meise in the TETTRIs project was look at the capacity in Europe for taxonomy. I think the same thing could be said for ecology capacity as well. We're focusing on, this project is about capacity on taxonomy, but that's taxonomy very broadly as well. It's about identification of things, not just primary taxonomy and to do with describing things. It's how taxonomists get involved in lots of different things. For invasive species, there are taxonomists who spend a lot of time trying to identify new things that come in. And of course, that's quite tricky when nobody's had any experience. None of those things are in keys in your language. And so taxonomists are important there as well. So you've possibly heard of the taxonomic impediment. This concept has been around for a long time. The fact we don't have enough capacity in taxonomy to even describe all the species we have before they become extinct. And up to now, all of the taxonomic impediment studies have been looking at the total number of species that are estimated for each taxonomic group and looking how many taxonomists we have for those particular groups. And I wanted to kind of look at this in a different way by more looking at how many taxonomists we have and how many taxonomists we need, so the demand. So if we accept we're never going to be able to actually describe all taxa that exist, we should perhaps have taxonomists more that address specific policy needs rather than just trying to tick a box. We already have so many taxa that only ever been seen once. There's an insect in a museum for everything and on a pin. But nobody actually can go out and do any monitoring for those because there are just so many of them. So taxonomic impediment is about limited capacity. And our study, we want to look at the geographic spread of taxonomists, the taxonomic coverage of taxonomists, and the link between supply and demand. Measuring taxonomic capacity of scale. So yes. So I'm a data scientist. Of course, my approach is to make a workflow. And we made a workflow looking at publication of taxonomic information because that's the most accessible way we can see about taxonomic capacity. So in a publication, you've obviously got the authors, you've got the affiliations of all those authors, and you've got the abstract that tells you roughly what the paper is about. So we can use those to find out lots of things about the authors. You can tell their sex, for instance. You can tell which country they were working in. You can tell, let me think, well, all sorts of things, but you can also look at the abstract and the title and figure out which taxa are involved in there. And we made this open and reproducible workflow to do this using OpenAlex, which is a open bibliographic database, Wikidata, and GBIF as well. So taxonomic capacity. So you have the absolute difference in the number of taxonomists in Europe for those different countries, and you have the per capita difference. So you can see a clear east-west divide in the number of taxonomists, and obviously bigger countries like Germany, the UK, and France are going to have more because they've just got more people. But you see there, they're not particularly high per capita compared to some of the other countries. When you look per capita, also a lot of this east-west divide disappears. And what this is really telling you, I mean, you've got Iceland there with also taxonomists for some reason. Move that. What this is really telling you is there's very little link between the number of species in this country and the number of taxonomists. It's more related to the number of people. And so that's a bit curious, isn't it? You would think it would be better to have more taxonomists in Turkey than Iceland, for example, because there's a very big difference in the number of species in those countries. It also means that small countries like those ones in the Balkans are never going to have enough taxonomists to be able to cover all of the species they've got. And so there really is a demand. And when you think of little places like Montserrat, even more so, that the bigger countries support taxonomy and the smaller countries, the more biodiverse countries, which does happen now. It has happened for a very long time, but for completely colonial reasons, that the bigger countries were trying to exploit all the smaller countries and the species in them. We kind of need, if we try and be uncolonial and not help those people, it doesn't help anybody. It doesn't help them. They're never going to have the capacity to do it themselves. We can increase their capacity to do it. We can help them increase capacity, but to some extent, because they have small populations, because they have very sparse populations and very high biodiversity, it's hard to imagine a situation where they can always do all the taxonomy themselves. If you look at the taxonomic capacity by species, there's no surprises here. So if you look at the plants at the top, this is the phylogeny of all plants. The vascular plants here, the Liliopsida and Magnoliopsida, they're the ones we eat. They're the ones that are in our gardens. Those are the ones we tend to be more interested in, and those tend to be the ones with the most research done on them. All the little algae, the single-celled stuff, the stuff in the sea and in the bottom of the river doesn't get much attention. If you look at the fungi, it tends to be the Ascomycota. Some of these groups are higher because they're lichens, and so they're more obvious. Soil fungi, probably not so much. And over here, I can't remember what groups those are, but they don't get much attention at all. In animals, biggest attention on insects. So they are the biggest group. Arthropods in general, but that mostly insects within arthropods. Some attention on things with a backbone. But any soft-bodied, non-arthropod, aquatic, invertebrate, yeah, not much interest at all. So probably some interest in some of those, the parasitic nematodes and things like this. But if you're an annelid in the sea, then probably not many people are interested. So, yeah, that's what you need to see there. So that was the demand side. That was the supply side, the supply of taxonomies. This is the demand side. So we took 9 different sort of policy drivers from Europe and looked at their taxonomic spread here. So for instance, a very obvious one is the birds directive. It's only birds, and so it's just that one little bit there. There's also pollinators, which is only a few groups of insects. But if you take this one here, this is one Helen did. It's a horizon scan for invasive species in Europe. You see a bit more of a taxonomic spread here. So they had some insects in there. So just if you're not a invasion biologist, these are species which are not in Europe, which are likely to spread in Europe. You're nodding, yeah. And they use experts to try and evaluate which ones are likely to establish and spread. So there's some chordates in there. There are some plants as well, but mostly vascular plants, but no fungi really to speak of. Maybe some mollusks, but there's some other groups there. Maybe none of those are invasive. It's hard to say, and it's also hard to get experts that are expert on this, and because we don't know what we don't know, and we don't know most about those ones. So if you take the supply of taxonomists and the demands from policy, and you try and model the number of taxonomists we have, including the number of species in each group, you do find adding the policy variables explains additional variation beyond species richness as a whole. So is that the taxonomists driving the policy, or the policy driving the number of taxonomists? Well, I think it's both. I think a lot of people are interested in birds. They show birds are declining. Policymakers pick that up. They drive policy on that. They may fund more work on birds. That goes back to the bird taxonomists. They make more evidence on birds. That goes back to the policies, and this cycles around. It's very hard if you're a marine worm to break into that cycle, or a marine worm taxonomist. So you can see it happening in things like the red listing. That red listing gets done on organismal groups that are well known, and lots of stuff ends up as being sort of data deficient or not assessed at all because people aren't thinking about marine worms at all. This is not necessarily a bad thing. Society has prioritizes things as it wants to, and it's good that policymakers are paying attention to taxonomists and taxonomists are paying attention to policymakers. That's not a bad thing at all. But in many respects, when you think about those two different groups, they tend to talk to each other through megaphones. So in as much as we publish papers that are not directed for policymakers, and somehow those policymakers need to pick up on that, or we try and put press releases out there so the press pick it up and the policymaker. So this is not a very good way of communicating, and the subtleness that maybe it might be worth having a project on marine worms or something like that gets lost in those sort of megaphone discussions. So no taxonomy, no defensible evidence. So taxonomy is important. It's a bit of a dry subject and it's not forgotten, but sometimes it's seen as stamp collecting when it really isn't. It is important. It's important to do for the identifications of things. If you want good trends on different species, we need taxonomy and good identification. All the legal reporting is done through taxonomic names. And particularly for things like early warning, we need good identifications on things. In the context of local communities as well, but everyone really, everyone wants to keep their data and use everybody else's data. And the term data sovereignty comes up over and over again. It comes up in the care principles as well. But if we're always completely sovereign, if we live on a desert island by ourselves and we have our coconut tree and it's our coconut tree, and someone else lives on a different island and they have a hammer, they could get into our coconuts, but their hammer is sovereign to them and our coconuts are our coconuts, and so they both starve to death. But so some cooperation is what humans do really well. It is great that people feel ownership of their data. If they feel ownership of their data, they maintain their data and they look after it, and they should have pride in their data. But yeah, I wrote this. So John Donne was a poet from the UK, and he has a line in one of his poets that no man is an island, and I'm sure he also meant no woman is an island as well. And he was not arguing against individuality, but arguing about interdependence of people. And I think if we want to do science evidence very well, we are all interdependent on each other. That's one of the reasons we should be sharing data and trying to do it in a very equitable way. And that goes true of indigenous people as well, because they can be completely data sovereign, but it doesn't really help them much. There has to be some sort of balance of sovereignty and sharing. So I should also mention artificial intelligence because it's impacting everything we do in science, but it'll also in biodiversity. We have a partner in One Stop Tokohoya who also coordinates the Mambro project, which has got these cameras you can drive around on cars, which identify invasive species by the side of the road. And we also have moth traps, which can identify moths automatically. And all the models built on these are built on the data we have at the moment. And although you can create massive amount of data, that you can also, there's also a great possibility of amplifying all the biases we already have. And so we have to be really careful that we're not just adding to the noise out there and the bias out there, because computers, well, I probably shouldn't tell you this, but I used to work for a company where I accidentally overpaid the pension schemes £5 million because of one very small mistake. My very small mistake gets amplified into a very big cost, and the company were very good about that. And we got the money back, but the pension schemes wanted compensation because they had invested it in the stock market, and the stock market had gone down in the meantime. So computers amplify mistakes, and that's the same as artificial intelligence does. And it's artificial intelligence, I guess, is also, because it's a bit of a black box, it's easy to hide those biases in as well. So yeah, I don't need to say more about that. So bring it all together. We struggle to turn biodiversity data into policy evidence because the data and contextual and the evidence and contextual. Most of our biodiversity data are unstructured. We have to address that, and we can't get around it. There's no alternative. Structured monitoring helps, but it still doesn't get us around the problem. We have the mismatches on the needs of different communities, and we still don't have all the capacity we need, and that good communication between the capacity we have in ecology and taxonomy and the capacity that policymakers need as well. So yeah, this just repeats what I said before, but perfect data are really available. Standardized monitoring can't answer all the questions. Much evidence can be built from existing data, and this is not a failure, but a constraint on what we have. We need to accept the heterogeneity, assess fitness for use, make both uncertainty and bias explicit, design work for reuse, support cooperation over pure sovereignty. And in this beautiful new world, we have repeatability. Our biases are not hidden. Local and global data become usable together, and policy decisions become more defensible. So we need more data, but that's not at all sufficient. Turning policy data into evidence also requires appropriate infrastructure, workflows, and cooperation. So for many questions about long-term change or large spatial extent, we simply don't have an option to wait for better data. We have to use what we've got. So thank you. I hope that was useful.